Whew, feels much better now. Today we'll discuss epistasis. Now I know you guys aren't biology majors, which is kinda lame, but I'll help you through it. Stasis is a type of gene interaction between two different genes. It usually involves one gene affecting another, most likely suppressing it or manipulating it. The gene that is masking or affecting another gene is called the epistatic gene, and the gene being affected is called the regulated gene. Today we'll be talking about one form of epistasis, which is dominant epistasis, which basically means the epistatic gene is masking the regulated gene because it is dominant. So single dominant epistasis is when a allele masks another allele because it is a dominant gene. So if we look at sunflower coloration, the original way a sunflower would go from the white precursor color, so its original color, to yellow would be through an enzyme or some genetic process. But with dominant epistasis, when there's a dominant A gene, it basically cancels out that enzyme, not allowing that white precursor to turn yellow. So it just still becomes white. Now I'm going to illustrate and explain two different crosses using single dominant epistasis, and I'm going to keep it real simple. So we can see that for the first cross, we have two dominant A alleles for each things we're going to cross. That means, basically, if there's a dominant A gene, so that's a big A, that means that the phenotype will always be white because the dominant A gene will always repress whatever the B alleles are doing. So if you see a dominant A gene or allele, it's always a white phenotype. Now if there's a recessive allele, A allele, then you have to look at the B phenotypes. So if the dominant is B, then it'll be yellow, and if it's a recessive B, it'll be green. If we do the dihybrid cross for the first one, we see we get 12 dominant A's, so white phenotypes, 3 dominant B's, yellow phenotypes, and 1 recessive B, green phenotypes, making the ratio 12 to 3 to 1. For the second cross, we do the same dihybrid cross, and we find that we get 8 dominant A alleles, making it a white phenotype, so 8 white phenotypes, and we get 8 dominant B alleles with no dominant A's, making it a yellow phenotype. So the ratio is 1 to 1.